Good evening, everyone. So, we'll start in 5 minutes so that more people can join in. Okay?
And this is the ultimate food tour. The breakfast of champions.
So, we can start our today's session. So, in the last class, we were looking at the basic revision of things, right? So, there we started about how exactly the MHC molecule is able to bind with the CD4, CD8 receptors and all those things. So we discussed about B cell receptor, T cell receptor. So we'll start with completing our revision. So we'll start with revising our T cell receptors. Okay. And today's class will be a little bit more theory heavy, because if you guys have seen Week 8 lecture, then you know that it was a pretty heavy discussion over how B cell actually matures and activates. Okay, so even our live discussion will be a little bit more heavy on the theory side. Okay, so starting with the revision of T cell receptor. So the summary of T cell receptor is as follows. In the adaptive immune system, the molecules responsible for specific recognition of antigens are antibodies and T cell antigen receptors. Antibodies, also called immunoglobulins, may be produced as membrane receptors of B lymphocytes and as proteins secreted by antigen stimulated B cells that have differentiated into antibody secreting plasma cells. Secreted antibodies are the effector molecules of humoral immunity, capable of neutralizing microbes and microbial toxins and eliminating them by activating various effector mechanisms. T cell receptors are membrane receptors and they are not in secretory nature. They don't have any secretory nature associated with them. The core structure of antibodies consists of two identical heavy chains and two identical light chains, forming a disulfide linked complex. Each chain consists of a variable region, which is the portion that recognizes antigen, and a constant region, which provides structural stability and, in heavy chain, perform the effector functions of antibodies. The V regions of one heavy chain and one light chain together form the antigen binding site, and thus the core structure has two identical antigen binding sites. So we are not discussing solely about T cell receptors, it is majorly in general receptor summary of B cell and T cell. Okay, T cell receptors consist of A chain and a B chain. Each chain contains one V region and one C region, and both chains participate in the recognition of antigens, which for most T cells are peptides displayed by MHC molecules. These facts now you must be knowing by heart, right? Because this is the thing which we are discussing and reiterating again and again since our first class. Okay. So we'll see a little bit more about this VDJ joining. So I have a small, video reference of this, how exactly VDJ joining works, and obviously textbooks are the best source. So you should go back to the textbooks, look at the VDJ joining, and then you'll understand how the variability is associated with the B cell receptors and T cell receptors. Okay, the V region of especially the immunoglobulins. Okay, the V regions of immunoglobulin and TCR molecules contains hypervariable segments, also called complementarity determining regions, which are the regions of contact with antigens. The inherited genes that encode antigen receptors consist of multiple segments separated in the germline and brought together during maturation of lymphocytes. In B cells, the Ig genes segments undergo recombination as the cells mature in the bone marrow, and in T cells and TCR gene segments undergo recombination during maturation in the thymus. Receptors of different specificities are generated in part by different combination of V, D and J genes segments. The process of recombination introduces additional variability in the nucleotide sequences at the site of recombination by adding or removing nucleotides from the junctions. The result of this introduced variability is the development of a diverse repertoire of lymphocytes, in which clones of cells with different antigen specificities express receptors that differ in sequence and recognition, and most of the differences are concentrated at the regions of gene recombination. During their maturation, lymphocytes are selected to survive at several checkpoints. Only cells with complete functional antigen receptors are preserved and expanded. In addition, T lymphocytes are positively selected to recognize peptide antigen displayed by self-MHC molecules and to ensure that the recognition of the appropriate type of MHC molecule, that is class 1 or class 2, matches the co-receptor. So for class 1 it will be CD8 positive T cell, and for class 2 it will be CD4 positive T cell, along with other co-receptors as well. Immature lymphocytes that strongly recognize self-antigens are negatively selected and prevented from completing their maturation, or in B cells undergo further light chain gene rearrangement that change their specificity, thus eliminating cells with the potential of recreating in harmful ways against cell tissue, or what this disease will be called autoimmunity, right? That our own cells, our own T cell receptor, B cell receptors are acting against our own body's antigen, our own proteins, and this will lead to autoimmune disease. So these checkpoints are pretty crucial to assure that the B cells and the T cells, which are allowed to stay in the system and are allowed to divide further, they are highly selective of the antigen, of the outside antigen, and not our own self peptides. So T cell mediated immunity activation of T lymphocytes. So how basically T cell is excuse me, mediating immunity. So there are two ways. First, T cell, whenever T cell will come, the pathogen type or the antigen type will be intracellular, because T cells are not able to see any antigen without the help of MHC molecule. Okay, and MHC can only process once the antigen is inside a cell. So imagine if phagocytic cell has engulfed that organism or the antigen. So what will happen? That organism might be able to survive at that point of time, and they might have escaped the lysosome and they are staying in the cytosol. So what will happen? At this time, the CD4 positive T cells that function by secreted cytokines in CD40L, which means the phagocytic cell will have CD40L receptor. This is ligand and the receptor will be here. And the other way can be via CD8 positive T cell that kill infected cell in which microbes have escaped from the phagosomes and are present in the cytosol. Second, if the genetic material of the pathogen is in the cytosol or inside the cell, what will happen? There is only one major way through which this case will be dealt with. It is with the help of CD8 positive T cell that will directly kill the infected cell, and they won't need any help of T helper cells. Okay, or particularly Th4 cells, CD4 positive T cells. And this was the overall view of how the cell mediated immunity works. So this is basically the summary of things, how the process happens. So once the antigen will be in the lymphoid tissue, the antigen will be recognized in the lymphoid organ. It can be via CD4 T cell and CD8 T cell. Now we know where CD4 positive T cell will help and where CD8 positive T cell will help. After this, the particular cells will be selected and will be allowed to proliferate and differentiate into the receptor which was effective against the pathogen. Once the differentiated effector and T cells they leave the lymphoid tissue, then they'll migrate to the place where the antigen is predominantly present, leading to the effector T cell encountering antigens in the peripheral tissue, and then this will lead to the activation of T cell, and once the T cell is activated, it will kill either by inflammation, enhancing phagocytosis, or directly killing the cell. In the same process, naive T cells, which are coming from the outside, majorly from the thymus. They'll enter and they'll help in making sure that the receptors are pretty specific with respect to the antigen, and this whole process will will occur in this way. These are the steps involved in the activation of T lymphocytes. So how a T lymphocyte will be activated? It will be activated once it sees the antigen along with the MHC receptor, and that way, once this selection is made sure that it is recognizing only the foreign antigen, the co-receptors are recognized, the co-receptor ligands are properly aligned, and then only it will be allowed to proliferate and differentiate. Okay, now this you very well know that how well the co-receptor are playing a major role in recognizing self and non-self. After spending almost 8 weeks with the course, now you know why we actually need so many proteins and so many surface receptors to make sure that the self and the non-selves are distinguished very well. So even if little bit of mishappening happens in the co-receptor recognition during the developmental process, it will lead to immense reduction in overall longevity as well as quality of life. Okay, why? Because if your cells or if your defense system is working against your own peptides, your own proteins, basically you won't be able to have a long sustained life. Okay, and you will have a lot of diseases. You need to depend on lot of external factors to survive. So all these checkpoints, however the selection is happening, however the cells are proliferating, however the cells are staying in the system, it is highly regulated. And once this regulation is off, then we know very few cases where the people survive basically, and even if they are surviving, the quality of life is not at all good. Okay, so here we discussed about how, again, this is the focused view of how TCR is basically recognizing the antigen and allowing the cell to act accordingly. Then these are few of the, I guess, I have the similar image, I guess, maybe in the coming slides. Okay. So here again, so when we talk about signaling, as I have mentioned earlier as well, the signaling is the way through which the cell is changing its biochemical properties, and it is the way through which the change is taking place or reaction is being done after an action. And here the action was recognition of an antigen. So we have an antigen, our cells know that there is an antigen. Now what to do? Okay, we have to decide whether we have to do inflammation, we have to proceed with enhanced phagocytosis, or we have to directly kill the cell. So all these actions will require intermediary steps which will do all these things, and that is dependent upon the signaling pathway which is being activated because of the receptor ligand conjugation. Okay, few of the signalings you can see, these are major signaling pathways. So it can be via RAS GTP, via PIP, via PI3 kinase, via calcium, AKT pathway, ERK JNK pathway, PKC pathway, calcineurin pathway. All these are basic signaling entities which are activated once the ligand is recognized by the receptor. And in immunology, we'll see once the antigen is recognized by the lymphocytic receptor. Okay. Then again, with respect to the T lymphocytic summary, we will start with the T lymphocytes are the mediators of the cell-mediated arm of the adaptive immune response, which combats microbes that are ingested by phagocytes and live within these cells or microbes that infect host cells. T lymphocytes also mediate defense against some extracellular microbes, help B lymphocytes to produce antibodies, and destroy cancer cells. The response of T lymphocytes consists of sequential steps: recognition of host cell associated microbes by naive T cells, expansion of the antigen specific clones by proliferation, and differentiation of some of the progeny into effector cells and memory cells. T cells use their antigen receptors to recognize peptide antigens displayed by major histocompatibility complex molecules on antigen presenting cells, which accounts for the specificity of the ensuing response, and also recognize polymorphic residues of the MHC molecules, and here by polymorphic means the co-receptors and the variants, accounting for the MHC restriction of T cell responses. Antigen recognition by the T cell receptor triggers signals that are delivered to the interior of the cell by molecules associated with the TCR and by the co-receptor CD4 and CD8. The binding of T cell to APC is enhanced by adhesion molecules, notably the integrins, whose affinity of their ligand is increased by antigen recognition by the TCR. APC is exposed to microbes or to cytokines produced as part of the innate immune reactions to microbes express co-stimulators that bind to receptor on T cells and deliver necessary secondary signals to T cell activation. The biochemical signals triggered by T cell by antigen recognition and co-stimulation results in the activation of various transcription factors that stimulate the expression of genes encoding cytokines, cytokine receptors, and other molecules involved in T cell responses. The TCR signaling pathways involve protein tyrosine kinases, which phosphorylate protein that become docking sites for additional kinases and other signaling molecules. The signaling pathways include the calcineurin impact, RAS-MAP kinase, and PI3 kinase mTOR pathway. In response to antigen recognition and co-stimulation, T cells secrete cytokines that induce proliferation of the antigen stimulated T cells and mediate the effector functions of the T cell. T cells proliferate following activation by antigen and co-stimulators, resulting in expansion of the antigen specific clones. The survival and proliferation of activated T cells are driven by the growth factor IL-2. Some of the T cells differentiate into effector cells that are responsible for eradicating infections. CD4 positive effector cells produce surface molecules, notably CD40L, and secrete various cytokines that activate other leukocytes to destroy microbes, and CD8 positive effector cells are able to kill infected tumor cells directly. Other activated T cells differentiate into memory cells, which survive even after the antigen is eliminated and are capable of rapid responses to subsequent encounter with the antigen. Naive T cells migrate to peripheral lymphoid organs, mainly lymph nodes, draining sites of microbe entry, whereas many of the effector T cells generated in lymphoid organ are able to migrate to any site of infection. This is important. So what it means is that naive T cells, the ones which have not yet seen the antigen and are selected, they'll directly go to the lymphoid organ, mainly lymph nodes, draining sites of microbe entry, whereas many of the effector T cells generated in the lymphoid organs are capable to migrate to any sites of infection. The pathways of migration of naive and effector T cells are controlled by adhesion molecules and chemokines. The migration of T cells is independent of antigen, but Cells that recognize microbial antigen in tissues are retained at these sites. So these are the basic characteristics of subsets of CD4 positive helper T lymphocytes. So there are multiple variants of CD4 positive T cell. It is Th1, Th2, Th17 and T follicular cells. So as you can see, their cytokines which are defining them, their cells on which the ligands are there or on which these particular effector T cells function, and their role in the diseases. Okay. Now the general properties of T cell cytokines. So why we need T cell cytokine, till now it is clear for you, and why we have so many different cytokines. You can see a little bit like small brief summarization of such cytokines, what are their functions and from which source these cytokines are secreted. Okay. Then again, as we have discussed, how basically both CD4 and CD8 cells, though we are studying them separately, but at the end of the day they both are working in cooperation among themselves to assure that the antigen is killed in the body. Again, this is just how the T lymphocyte, so I have a little bit better diagram with respect to this, that how basically T cells are going to the peripheral system. But one thing which you should remember from this is the receptors and the adhesion molecules which are taking place in ensuring that the cells are moving towards the source of infection or going towards the periphery from the lymphoid site. Okay. Then this is basically the ways through which evasion of cell mediated immunity by microbes. So we know that in some cases the microbe might evade our own system because we fell sick, right? So even having all these checkpoints, all these soldiers for us to prevent us from the disease, some of the microbes, they either escape or their sheer number is too much that our own system is not able to manage them in the proper time before we start to see any symptoms. Okay, but our immune system is constantly working. So such summary is given here. So you can see these are renowned microbes which are known to escape immune system, and there are loads of studies on them, and we actually use sometimes how they are evading the immune system. We try to use their mechanism to ensure that our foreign gene or our foreign peptide, which nowadays are being used as medicine or drug, can help the patient without being targeted by the immune system. Then the overall summary says, cell mediated immunity is the arm of adaptive immunity that eradicates infections by cell associated microbes. This form of host defense uses two types of T cells: CD8 helper T cells recruit and activate phagocytes and other leukocytes to kill ingested and some extracellular microbes. The CD8 positive CTLs eliminate the reservoirs of infection by killing cells harboring microbes in the cytosol. CD4 positive T cells can differentiate into subsets of effector cells that make different cytokines and perform distinct functions. Effector cells of the Th1 subset recognize the antigen of microbes that have been ingested by macrophages. These T cells secreted interferon gamma and express CD40L, which function cooperatively to activate macrophages. These are all the same things. Okay, it's just we are writing them so that we have better understanding of the sequences, because via images you can understand the process, but at the end of the day you need vocabulary to build the story around it. Okay, classically activated macrophages produce substances including reactive oxygen species NO and lysosomal enzyme that kill ingested microbes. Macrophages also produce cytokines that induce inflammation. Th2 cells stimulate eosinophilic inflammation and trigger the alternative pathway of macrophage activation, and T follicular helper cells induced in the parallel trigger IgE production. So we'll see this in the coming slides what exactly it means. IgE and eosinophils are important in host defense against helminthic parasites and in allergic diseases. The balance between activation of Th1 and Th2 cells determine the outcomes of many infections, with the Th1 cells promoting and Th2 cells suppressing defense against intracellular microbes. Th17 cells stimulate the recruitment of neutrophils and monocytes that destroy extracellular bacteria and fungi. Th17 responses also underlie some chronic inflammatory diseases. CD8 positive T cells differentiate into CTLs that kill infected cells mainly by inducing apoptosis of the cells. CD4 positive and CD8 positive T cells often function cooperatively to eradicate intracellular infections. CD8 positive CTLs also kill cancer cells and are the principal mediator of anti-tumor immunity. Many pathogenic microbes have evolved mechanisms to resist cell-mediated immunity. These mechanisms include inhibiting phagolysosome fusion, escaping from the vesicles of phagocytes, inhibiting the assembly of class 1 MHC peptide complexes, producing inhibitory cytokines or decoy cytokine receptors, and inactivating T cells, thus prematurely terminating T cell responses. Okay. So the tolerance basically means that what sort of antigens are seen by the T cells. So if we look at the central tolerance, this is basically in the lymphoid organs, that is thymus and bone marrow, while the peripheral tolerance, as the name suggests, is in the tissue side. Okay, so in the central tolerance, the lymphoid, the common progenitor cell, will lead to the immature lymphocytes. Once these immature lymphocytes recognize the self antigen, they'll be led to not survive. Okay, they'll be killed, or if the binding was good enough or not so good enough, then a little bit of more rearrangement will be done, editing will be done, and regulatory T cells will also be there to ensure that this function is happening properly, or this whole process is happening good enough. And in the peripheral tissue, when these cells migrate and when they recognize the self antigen, if they are binding with the self antigen very strongly, they'll be subjected to apoptosis. If they are binding self antigen poorly, very poorly, then they'll be allowed to stay there, but little bit of rearrangement needs to be provided, or needs to occur. If the receptor is not at all sensitive and they are dimerizing, then the receptor is not good enough and the cell will enter into energy where the cell is not recognizing any antigen. And if the cell is recognizing the self antigen moderately, it will allow to stay and it will further be selected for proliferation. Okay, so basically we'll talk about the central tolerance and the peripheral tolerance in a little bit more detail now. So central tolerance is the negative selection. So it occurs in the thymus during T cell development. Immature T cells with TCRs that strongly bind self antigen or MHC, they'll undergo apoptosis, because a cell should not bind to our own antigens. This will ensure self reactivity. T cell die before entering the circulation. It affects CD4 positive T cells and CD8 positive T cells. Then when we talk about epithelial cells and autoimmune regulators, so MTECs, that is medullary thymic epithelial cells, express in many tissue restricted antigens, while autoimmune regulators induce expression of these antigens, allows T cell to be tested against a wide variety of self proteins. Then it will lead to, again, there's a whole sequence of things that goes under negative selection and positive selection. Okay, if we talk about Treg development or T regulator cells, so some CD4 positive self reactive thymocytes, they don't die, instead they become regulatory T cells. So T regulatory migrate to lymphoid organ or peripheral tissue, suppress activation of any remaining self reactive T cells. So, and let me complete this first. Add an extra layer of self tolerance beyond central deletion. So imagine, even after central tolerance, if some cells by chance they are showing, because they are seeing new antigens in the periphery, self antigen as well as foreign antigen, and if there are cells which are recognizing self, or even if they are not recognizing self, but after the immune response, these cells need to be taken care of because their role is done now, their function is over. So we cannot keep sending more and more lymphocytes to the periphery, right? So in that case, the regulatory T cells actually help. Then why it is needed, the clinical insight will be: negative selection is imperfect, some self-reactive cells do mature. Okay, and this is a very crucial balance between selection, and we don't understand completely this process. Still a lot of research is happening in this area to get all the key players which are involved in the selection process. The peripheral tolerance mechanisms are essential to prevent autoimmunity. Okay, then if we talk about, so this is a completely new topic. So the phases and types of humoral immune responses. So in humoral immune responses, what are the different phases? So humoral immune response means via antibodies. Okay, so the activation, obviously the first thing will be the activation of B lymphocytes. This leads to proliferation or clonal expansion, which we have seen in details, and differentiation into plasma cells that secrete antibodies. Naive B cells express IgM and IgD as membrane bound receptors. Activation require antigen binding and additional signals. Antibody secretion. Secreted antibodies have the same specificity as the B cell receptor that recognized the antigen. Once activated B cell can produce thousands of plasma cells, each secreting thousands of antibodies per hour. Class or the isotype switching. So some B cells change antibody heavy chain class, that is IgG, IgA, IgE, enabling different effector functions against different microbes. Affinity maturation. During the immune responses, antibodies improve their binding strength to antigen, enhancing their ability to neutralize microbes and toxins. Antibodies responses are T dependent. They basically need T helper cells, or T independent. They do not need any T helper cell for their responses. In T dependent response, it is triggered by the antigens, require helper T cells which recognize the same protein antigen via MHC, lead to class switching, affinity maturation, long-lived plasma cells and memory B cells. They produce strong, specialized and long-lasting antibody responses. In T independent response, they are triggered by non-protein antigens such as polysaccharides, lipids, nucleic acids, multivalent antigens. Okay, do not need T cells. These B cells are directly activated with innate signals. Responses are usually short-lived, weaker, and lack class switching or affinity maturation. If you talk about follicular B cells, they resides in lymphoid follicles and recirculate, mediated in T-dependent response to protein antigens, produce class switched high affinity antibodies, and give rise to long-lived plasma cells. Marginal zone of B cells, we are talking about the lymphoid organ, lymph node, and especially the germinal center, found in the splenic marginal zone and lymph node outer follicle rims, respond mainly to blood-borne polysaccharide and lipid antigens, produce T-independent IgM antibody with limited receptor diversity. B1 cells, located in mucosal tissues and peritoneum, respond mostly to multivalent polysaccharide and lipid antigens, also produce T-independent IgM antibodies, can secrete natural antibodies without immunization that clear apoptotic cells and provide some protection against bacteria. Overall, follicular B cells, they are T-dependent, high in affinity and long-lived, while the marginal zone and the B1 cells which are there, they are T-independent, majorly IgM, which is not that diverse, and natural antibodies. When we talk about primary antibody responses, so all these things will be little bit more clear because I have their representation in the coming slides. Okay, so the primary antibody responses, this happens when there is first exposure of an antigen to the system. Okay, this graph we have already seen. It's now that we are looking at into from a theoretical point of view. Lag phase, slow onset, usually takes 7 to 10 days before detectable antibodies appear. Antibody quantity, so generally it's low, fewer plasma cells are generated, so antibody levels are modest. Antibody isotype, mainly IgM, which is the default B cell receptor, little to no class switching in early stages. What about affinity? Low, and affinity is also low, so antibodies have not undergone much affinity maturation, so binding to antigen is relatively weak. This is the initial time, so the body is still adapting to the newly found antigen, but it will do whatever is required to control the spread of the infection. Okay, memory cells, some memory cells are generated but their numbers are limited. Duration of primary antibody response, the response is short lived, antibody levels decline relatively quickly. Now we talk about the secondary antibody response. This happens when the second time the antigen is coming to the system. So the lag phase in this, it is like visually not present, so the lag phase is not there, so the response is rapid. Okay, rapid onset antibodies appear within 1 to 3 days due to the presence of prior memory B cells. The quantity of antibody, so in comparison to the primary response, the antibody number is much higher, and the cells are also, or the plasma cells are also in huge number. Antibody isotypes, so predominantly in the primary response it was IgM, but in secondary response it is more specific, that is IgG, with class switching to IgA and IgE depending upon the context or depending upon the antigen. IgM still produced but to a very smaller extent. Affinity, So as the antibodies are highly specific, the affinity is also high. Due to repeated affinity maturation in germinal centers, antibodies bind antigen more strongly and effectively. Memory cells, so large pool of memory B cells are ready and they are quick to respond. Duration, this is generally long lived, and antibodies levels remain elevated for longer, providing longing and lasting immunity. So if we see the key features and the responses, so if we talk about exposure, so in the primary response, the system is seeing the antigen for the first time. The secondary response occurs when there is subsequent infection. What is the onset or the lag phase? So primary response is a little bit slow, so around 7 to 10 days, while secondary response is quick and it is between 1 to 3 days depending upon the antigen. Antibody quantity, so in the primary response the quantity of the antibody is less. In subsequent response or the secondary response, the antibody quantity or antibody number is more. The isotype which is present in primary response or primary response against the antigen, it is IgM, while in the secondary response it is IgG, and then depending upon the antigen it can be IgA and IgE. Affinity in the primary response is low, while it is highly specific and more accurate in secondary response. Memory cells, in the primary response few of the plasma cells will be there, and in secondary response there will be large memory cells. Duration, the primary response is short lived, while the secondary response is long lasting and it will provide enhanced immunity. Now this is a very if you guys have attended this week's session, you know this week was all about B cell activation, differentiation, so basically development of B cells. Okay, so this is the overall view of B cell development. During the antigen independent maturation phase, immunocompetent B cells expressing membrane IgM and IgD are generated in the bone marrow. Only about 10% of the potential B cells reach maturity and exit the bone marrow. Naive B cells in the periphery die within a few days unless they encounter soluble protein, antigen and activated Th cells. Once activated, B cell proliferate within secondary lymphoid organs. Those bearing high affinity differentiate high affinity Ig bodies or immunoglobulins differentiate into plasma cells and memory B cells, which may express different isotypes because of class switching. The numbers cited refer to the B cell development in the mouse, but the overall principle apply to humans as well. So you can see here very well that the development of B cells occur in the bone marrow. Okay, there are two ways through which they can develop further. It can be in thymus dependent or thymus independent. So when we are saying thymus dependent, which means they need T helper cells, and when we are saying thymus independent, which means they do not need T helper cells and they can be activated without the help of T helper cell. And how they can be activated? Because they are also APCs, remember, antigen presenting cells, which means B cells can see the antigen and then their activation can be done depending upon the location, okay, or whether the T helper cell was available or not available. If we look here, this is the bone marrow. there is no antigen in the bone marrow. So these are naive cells or pre-cells, say. They are not yet mature. Once the progenitor B cell is there, it will undergo heavy chain rearrangement, right? And multiple rearrangement will happen for the receptor, and then it will be mature B cell. This mature B cell will then come to the peripheral organ or peripheral lymphoid organ. Here in peripheral lymphoid organ, you know there will be follicular zones or dark zone, okay, of germinal center. So we are talking about this, that when the B cell or the mature B cell who has rearranged heavy chains, who has preformed light chain as well, now it enters the peripheral lymphoid organ. Here, if it is not able to recognize or find any antigen, it will be cleared from the system by apoptosis, because the receptor which was there on the B cell, it is of no use. There is no antigen for it. Okay, but if this naive B cell recognize the antigen and also T helper cells are present, now this B cell, the mature B cell which entered, it will be activated. And by activated, we means the type of antigen it recognized and switching the class to secrete the antibody. The antibody will just recognize the antigen, it will happen here, and then the plasma cell will basically secrete the antigen, the antibody, and also the memory B cell will also be formed and stored there, okay, in the peripheral lymphoid system. So this is the overview of how the B cell basically matures with the help of T helper cell. And now we have already discussed the key players. So for example, if T helper cell, how the T helper cell will help B cell maturation? They'll provide cytokines, okay, because T helper cell is helping in the activation of B cell. Now, as I have mentioned earlier, there are T dependent and T independent antibody responses. So if we see the overview of T dependent type of antibody response, so in T dependent, the follicular B cells which we just saw previously, the follicular B cells will have IgM, which is a surface receptor of a B cell, and it is not secretory in nature. It will directly see the antigen or the protein antigen. It will go and find T helper cell, or the T helper cell will come and bind with this activated B cell, and then it will lead to the antibody class switching, which means more high specific and highly effective antibodies will be formed from this activated B cell. Okay, while in T independent type of antibody release, so this type of cell directly saw the antigen, or say the outside of the antigen. It doesn't need proteins for it, not necessarily. Okay, then what will happen? This will lead to the complement activation, which we have not discussed. We'll discuss it soon, because it's in itself a very huge topic to discuss. Okay, then it can be complement proteins, it can be other ligands. Then it will directly switch from IgM secretory B cell, the IgM cell, to plasma cell which just secreted the IgM. So these type of antibodies, they are short lived, they are not highly specific, because you just see, right, what they recognized. They recognize the polysaccharide, not the protein, which is the processed antigen. So this is the initial way of controlling the infection. This will be short lived, as we have seen here, right? In the primary response, the affinity is low, memory is very less, the duration will also be very short. Okay, and so it is not highly specific, but it will provide the body some time to get into more specific ways to target the infection. Now if we talk about the primary and secondary antibody responses, which we have just read in the previous class, this previous slides. Just give me 1 minute, I need to have a little bit of water. Sorry. So whatever we have read in the previous slides, we are seeing them in the visual format now. So in the primary response or the primary antibody response, it will take around 7 to 10 days. Why it is taking 7 to 10 days? Because the naive B cell, which by default will have IgM, it will see the ligand see the antigen, then it will be activated, and by activating means the process of class switching and the process of affinity maturation will take place. After this, it will release the IgM and it will control the limited antigens which are there, and also some of the memory B cells will also be formed. But the number of memory cells is low, but the memory cells are there now, right? Earlier it was 0 if we talk about memory B cells. Now once the exposure or the infection happens again, then the memory B cells will take over from here. We don't need any more naive B cells. Okay, we have memory B cells now. These memory B cells will proliferate and will start secreting more effective, more specific antibody, and in the secondary response primarily it will be IgG. And in a basic connection problem. So where were we? So we were discussing about the features of primary and secondary antibody responses. Okay, so primary and secondary antibody responses, they differ in several aspects. Okay, so in a primary response to any infection or say vaccination, naive B cells in secondary lymphoid organs are activated to proliferate and differentiate into antibody secreting plasma cells and memory cells. Okay, so this we have seen since the beginning. Okay, and when these memory cells, they are there, this is the reason why the secondary response, there is no lag phase associated with, little bit, but it's lesser than the primary response. Okay, so some plasma cells may migrate to and survive in the bone marrow for long periods of time. In a secondary response, memory B cells are activated to produce larger amount of antibodies, often with more heavy chain class switching and affinity maturation. These features of secondary responses are seen mainly in response to protein antigens, because these changes in B cells are stimulated by helper T cells, and only proteins activate T cells. Okay, so and also the kinetics of the response may vary with different antigens and type of immunization. So all these things are pretty diverse, right? But at the end of the day, the basic principle remains same. The intensity of these responses can change. So if we are saying that in the secondary response only the memory B cells they are activated and then they start to class switch and then they secrete antibodies, right? So this is happening with the help of helper T cell. Now we have studied about the helper T cells, how they help B cell and all that stuff we have seen. So now you have to bring your concepts to a single point, that whatever you have studied you should culminate them at one point when I am saying activation of B cell. So whatever we have studied for the activation of B cell will happen once the secondary response is happening. And what will happen in the secondary response? That there will be very small lag phase, there will be class switching, there will be antibody secretion and all those things. Okay, and if we look at like the basic features of primary response and the secondary response, or say differences of primary response and secondary response. So after first infection slash vaccination, so the lag phase after immunization in primary response is 5 to 10 days. Now you know why. Okay, while in secondary response it is usually 1 to 3 days. The peak response is smaller. Okay, so you can see the intensity as well. The amount of antibody is smaller in primary response or less in comparison to the secondary response. And the isotype in the primary response is majorly IgM, while in secondary response there is more of IgG. And then again, depending upon the infection type of immunization, there will be IgA or IgE. It completely depends upon the type of infection, but predominantly in the primary response there will be IgM predominantly, and then in secondary it will be IgG and other antibodies depending upon the type of infection. And the affinity of the antibody, so it's lower average affinity, more variable. Okay, because it is not specifically meant for the antigen, we need more time. Okay, and then for the secondary response the antibody affinity is higher or more, definitely, because it will be more effective, more specific, hence the affinity will be more. Okay, then if we talk about the antigen receptor mediated signal transduction in B lymphocytes, so since the beginning I am saying why do we need signaling. So signaling is the only way through which, when we say oh the cell is communicating from the outside world to the interior of the cell, how it is communicating. So these are the basic signaling systems, biochemical reactions, changes of the cell interior once it is under some influence. And here the influence can be binding of ligand to the receptor, or binding of say any ligand molecule to the receptor, or anything, a basic change, which that change it needs to be sent inside the cell so that the appropriate measures can be taken place. Okay, so cross-talking of antigen receptors on B cells by antigen triggers biochemical signals that are transduced by the immunoglobulin associated proteins Ig alpha and Ig beta. These signals induce early tyrosine phosphorylation events, activation of various biochemical intermediates and enzymes, and activation of transcript factors. Similar signaling events are seen in T cells after antigen recognition. Maximal signaling requires cross-linking of at least 2 Ig receptors by antigen. Okay, so there are a lot of abbreviations used. So for example, AP1, the transcription factor AP1 is called activator protein 1. GDP and GTP, so GDP is guanosine diphosphate, GTP is guanosine triphosphate. ITAM, so this you can see is a membrane receptor, so ITAM is basically immunoreceptor tyrosine based activation motif. NFAT is a type of transcription factor called nuclear factor of activated T cells. So from the abbreviations as well, you can get little bit of idea what these factors might be doing, right? And then NF kappa is nuclear factor kappa B, and PKC is protein kinase C, PLC is a phospholipase C. So these are standard molecules which are present in all the cells, but how they are being activated, it is the thing which changes the chemistry of that particular cell. Okay, how they are being induced, they change the way the cell will adapt. So if we see the cross-linking of membrane IgG by antigen, it's basically immunoglobulin. This can be any antibody, right? Any B cell receptor basically. So the cross-linking of membrane Ig by antigen. So the antigen has just bound. Then what will happen? This will lead to some structural changes, and here majorly it is tyrosine phosphorylation event. So once the immunoglobulin binds to the antigen, it leads to the change in the this ITAM receptor, which phosphorylate. Earlier it was not phosphorylated, later it gets phosphorylated. Then it gets recognized by other adapter proteins, which recognizes the phosphorylated receptor. Okay, once the adapter protein recognizes a phosphorylated receptor, then multiple secondary signaling molecules or messenger molecules are there in the cell, which are there to help in transmitting the signals which they just received from these receptors. So it can be via here in this particular description, it can be via PLC, it can be via RAS-RAC complex. If it is via PLC, then the downstream signaling for PLC is inositol triphosphate, which basically means that there will be increased cytosolic calcium ions, and once the calcium ions are there, then the calcineurin enzyme, which was already present in the cell but it was not activated, it gets activated whenever there is more calcium in the cell, and once the calcineurin enzyme is activated, then it will regulate the NFAT transcription factor. Once this NFAT is there, it will go to the nucleus and then translates the respective genes to which it will bind. And once the genes are being translated, then a protein will be here, and again this is the change, basically the biochemical change. So this adaptation or this reception of ligand led to the activation of certain genes, which will either make a new protein, inhibit a new protein, it depends upon the context. Okay, then second, if again, so PLC can act via increasing the cytosolic calcium ions, as well as it can also act via DAG, that is diacylglycerol. If diacylglycerol or DAG is present in the system, it will activate PKC enzyme. Once the PKC enzyme is activated, just like NFAT, it will work with NF kappa beta. It's a transcription factor. It will do again the same thing. It will go to the nucleus and it will transcribe and translate proteins, and hence the change will come with this particular cell. Now again, if we talk about GTP RAS-RAC pathway, then it will activate RAS-RAC GTP pathways, which will in return will activate ERK and JNK enzymes, and the transcription factor associated with ERK and JNK enzymes are AP1. These are few of the transcription factor, major transcription factor, which are predominantly present with Ig or in B cells. Cells having Ig receptor or immunoglobulin receptor or B cell receptor. Okay. Now next we had talked about the mechanism of helper T cells mediated activation of B lymphocytes. So at the end of the day, B cells are there, receptors are there. We saw if the ligand is binding to them, what happens inside the cell. Now we are saying the cell is there. Now it is how it is basically connecting with the T cell to let the biochemical process takes place. So here the antigen was microbe. Here the antigen, the recognition of the antigen, say not the antigen but say the counterpart is the T cell. Okay, so not only the receptor or the B cell receptor needs to be recognized by the T cell, there are other co-receptor molecules which are associated with this interaction to make sure that only our cells is being activated. These are the checkpoints which will assure that we are not going overboard, that it's not a faulty interaction, that it is not any internal cell which is which are playing tricks on us. So these interactions you need to remember because they are important, and they are these interactions, co-receptors, is the way through which a cell recognizes self. Okay, so if I give you an example, so for example, how would you recognize your say your family member or say your friends, right? So there's some image of that person in your head. When that person fits that image well, then you'll say, oh, this person is my friend. And imagine someone is closely related to that person comes there, then you get little bit confused sometimes that oh, is he my friend or not? And then you quickly come to a conclusion, no, he's not my friend, he's a doppelganger. Okay, so this is exactly what cells are also doing. They are just trying to avoid any foreign cell or any non-self cell to be recognized as self cell. So to make sure that the cell which they are connecting with is our own cell, so there are certain features and cells, how they talk via receptors and ligands. So there are certain receptor ligand complexes which are present on both the cells through which they'll recognize that, oh, this is my own cell and now I can talk to this cell. Okay, so some of these interactions which are important is when helper T cell recognizes the peptide antigen presented by B cells, the helper T cells are activated to express CD40 ligand. Remember, it's the ligand, and secrete cytokines, both of which binds to their receptor on the same B cells and activate the B cell. Okay, so on the B cell, what receptors need to be there? It needs to be cytokine receptor and CD40 receptor. So if the T cell is secreting cytokine, it should reach the respective B cell. How it will reach? Whenever there is a receptor for it. If there is a ligand on T cell, CD40 ligand, the receptor should be on the B cell, CD40 receptor, so that they can bind, as you can see in the middle panel here. Okay, and then this cell will again go to class switching and then it will secrete the antibodies. Okay, depending upon the type of antigen. So these are just the basic things which we saw here. The same thing, similar thing, not same thing, similar thing will happen here when this ligand will bind to the receptor. So this release of cytokine is a change or adaptation in the T cell. So once CD40 receptor binds to the CD40 ligand, in T cell now some biochemical processes might be taking place which will lead to the secretion of cytokine. So imagine there will be a transcription factor which will go and bind to a gene which is required for the secretion of cytokines, or a protein which is required for the secretion of cytokines. Okay, this is how the whole process is taking place. These are the basic things of receptor ligand complex. Okay, next, now we are talking about B cell making antibodies, memory B cells. So where exactly these things are happening? So these things are happening in the lymphoid organs, especially in the lymphoid organs, the germinal center. So B cells that have been activated by T helper cells at the edge of a primary follicle migrate into the follicle and proliferate, forming the dark zone of the germinal center. Germinal center, B cells in the dark zone mature their Ig genes at an extremely high rate, a process called somatic hypermutation, and migrate into the light zone, where B cells with the highest affinity Ig receptors are selected to survive, and they differentiate into plasma cells or memory cells, which leave the germinal center. The right panel, this one, shows the histology of a secondary follicle with a germinal center in a lymph node. So you can see the dark and the light zones majorly. So this is the this is the follicle, this is the germinal center, this is comparatively darker than this, right? So this is the dark zone, this is the light zone. So the germinal center includes a basal dark zone and an adjacent light zone. If we look at the overview of how cells are there in the germinal center and how they mature at the end of the day, and here by maturation I mean that they are getting converted into plasma cells and memory B cells. So plasma cells secreting antibodies and memory B cell getting stored, so to make sure that during the secondary infection the lag phase is not there and the defense will be more accurate, specific, and with higher intensity. Okay, so once the B cells are activated, so how they will be activated? They will be activated by the helper T cell. How they will be activated by the helper T cell? This is what we just saw, with the help of co-receptors and specific cytokines. Okay, once this is done, it will, so the changes which will happen after the helper T cell is helping B cell. So why there is need of helper T cell? So B cell needs help in doing what? Help in proliferation, help in maturing, okay, help in basically secreting the specific antibodies. So in the dark zone of germinal center, the B cell will proliferate at extremely high rates after getting help from the helper T cell. Once they are there in number, then what will happen? Somatic hypermutation will happen, and in somatic hypermutation, basically they mutate their Ig genes. So if you remember, we have seen the formation of VDJ joining and all those specificities, non-specificities, all those things will come into play. So somatic hypermutation and affinity maturation will take place. This will ensure that whatever the antibody we are making now is highly specific type. So it can be IgG1 or IgG2, right? So those things will be specified at this zone. Once they are there, then to make sure that whatever the antibody has been formed, it is of higher affinity, so what is required, and then it will exit the germinal center and they'll go wherever they are required or wherever they are needed. Okay, so here again the checking will happen. So by checking, it basically means that it should not recognize self. It should only recognize the foreign antigen, which is shown here, foreign antigen with higher specificity, and then these antibodies will be secreted into the serum, and this we talked about here. Once the antibody is there in the serum, then you will get all these responses, the primary response and the secondary response. Okay, I hope this makes a little bit of more sense now when we are revising it and going through it. Now, immunoglobulin heavy chain class isotype switching. So what do we mean by when we are saying that this affinity maturation will take place or specific antibodies will be made? How will be they made? So again, anything which is being made by the cell will need specific signals or specific signaling molecules to make sure that that particular work is happening. Okay, so in the B cell, so imagine this is the B cell, or say it is the antigen stimulated B lymphocyte, and it may differentiate into IgM antibody secreting cells, or under the influence of CD40 ligand and cytokines, some of the B cell may differentiate into cells that produce different Ig heavy chain classes, which are called isotypes. The principal effector functions of some of these isotypes are listed here, and we'll go through them. All isotypes may function to neutralize microbes and toxins. B cells activating factor belonging to the TNF family is a cytokine that may be involved in switching to IgA, especially in T-dependent responses. Switching to IgG subclass is stimulated by the cytokine interferon gamma in mice especially, but in humans it is thought to be stimulated by other cytokines. So as I mentioned earlier as well, that these things are still under research. We don't know much about how the selection is happening, how the class switching. We know the process, but there's still a lot of things which are not yet crystal clear that exactly this will happen. Okay, so imagine this is the B cells, and with the help of helper T cells and CD40 ligand and cytokines, which we just saw in the previous slide, it can go into say different types of Ig molecules. So for example, if it is turning into IgM or secretory IgM, okay, so it will be done, or the effector of IgM will be the complement activation, which we have not yet covered. And then if there is Ig subclass, so there will be multiple Ig, it can IgG, it can be 1, 2 and all those things, right? So once the IgG subclass is there, the primary function or the principal function of IgG, and remember IgG is majorly coming in the secondary response, okay, so it will be the Fc receptor dependent phagocytosis and phagocyte activation, complement activation, and neonatal immunity. Basically this is the subclass which is transferred via the placenta to the baby. So remember, the babies have innate immunity, right, and also the adaptive immunity, passive adaptive immunity, because these are the antibodies which are given by mother to the fetus or to the baby. Okay, if IgE is there, so basically if the B cell receives interleukin 4 and 13, it will convert into IgE secretory plasma cells. So the function of IgE is mast cell degranulation, and so this happens when you have immediate hypersensitivity. So remember, you get I don't know whether you guys have faced this or not. So basically you touch something and immediately your skin gets inflamed, it's red, itchy. So those are hypersensitivity to certain molecules, right? So in that case, which antibody is taking place? It is the IgE type of antibody. Okay, and when IgA will be formed, so when IgA type of antibody is there, so majorly B cells might have gotten these signals. So these signals are TGF beta, BAF, and multiple other things are as well are also present. So in IgA, if IgA antibody is there, and remember IgA, IgE and IgG, these are all predominantly for the secondary lymphocyte, secondary response. Okay, so whenever IgA is there, then mucosal immunity, transport of IgA through epithelia, so all those things will be there if IgA antibody is present. Okay, then this is the affinity maturation in antibody response. So by now you know what affinity maturation is. So in somatic mutation in Ig, V genes, selection of high affinity B cells. So basically early in the immune responses, low affinity antibodies are produced. Okay, during the germinal center reaction, somatic mutation of immunoglobulin V gene happens, and the selection of mutated B cells with high affinity to antigen receptor results in the production of antibodies with high affinity for particular antigen will be selected and will then be released in the serum. This process we have seen, and we might see in the coming slides as well. Okay, then this is the selection of high affinity B cells in germinal center. So this is what we said. This is a simplistic representation. We just say, oh, low affinity antibody was there, and then there was somatic mutation, and then there is high affinity. But exactly how this high affinity antibody is being selected. So remember, naive, 1 minute. Yeah, so initially a naive B cell was there. It sees an antigen, so the B cell activation by protein, antigen and helper T cell will take place. So whatever we have studied after the primary secondary response, it is a culmination of it. Okay, so some activated B cells migrate into follicles to form germinal centers, where they undergo rapid proliferation and accumulate mutations in their immunoglobulin V genes. These B cells produce antibodies with different affinities for the antigen. B cells that recognize antigen bound to follicular dendritic cells, antigens are selected to survive. FDCs or follicular dendritic cells display an antigen by using FC receptors to bind immune complexes, or by using C3 receptor to bind immune complexes with attached C3b and C3d. So these are complement terminologies, don't worry about them. Once B cells bind the antigen, process it and present it to the follicular helper T cell in the germinal center, and the signal from the T follicular helper cells promotes survival of the B cell. And as more antibodies produced, the amount of available antigen decreases. So only the B cells that express receptor with higher affinities can bind the antigen, and they are selected to survive. Okay, so this is the basic summary of developing and selecting high affinity B cells. Okay, so the cells will be there, we have seen it. So the B cells which are having low affinity will undergo somatic mutation, and once the somatic mutation take place, there will be rearrangement of the heavy chain. Once the heavy chain is rearranged, it will see the antigens which are presented there with the help of follicular dendritic cells. The cells which are binding to it with high affinity, they will be selected with the help of follicular helper T cell, and then they will be allowed to proliferate basically, and then secrete the specific antibody, and all other cells which are not selected will be subjected to apoptosis. Okay, so I guess we can go to some of the questions now. Let me get, I think, yeah. So as usual, you will get 1 minute each for the questions. So the first question for today is, it's a pretty easy question. So the first question for today is, T cells and B cells share common place of development, origin, set of surface receptors, or all of the above. So your 1 minute starts now. Yeah, so the right answer is origin, right? So B cell and T cell share common origin. Next question is, during development, if the T cell receptor interacts with MHC1, then it would become a CD8 positive T cells, CD4 positive T cells, double positive T cell, or CD4 negative T cell? This you guys should answer within like 5 seconds, but still we'll have 1 minute and your time starts now. Yeah, so your 1 minute is over. The right answer is CD8 positive T cells, right? I don't think so that I have to repeat or explain this anymore. So next question is, what is the correct sequence of T cell activation in lymph nodes? Okay, so the first is activated T cells differentiate into effector T cells and exit from the lymph node. T cells enter lymph node from blood via HEV. T cells not activated by antigens exit the lymph node. Activated T cells lose the ability to exit from the lymph node. Choose the correct option. So you have to choose the sequence. Okay, and it's a little bit bigger question. I'll give you guys 2 to 3 minutes, but like definitely less than 3 minutes. So think of all the sequences which happens and then try to come up with the right answer. Your time starts now. Your 1 minute is over. I guess we can see the answer, right? So the right answer is C. So basically the first thing that will happen is the T cells enter lymph node from blood via HEV. Then T cells not activated by antigens exit the lymph node. Then activated T cells lose the ability to exit from the lymph node. And then finally activated T cells differentiate into effector T cells and exit from the lymph node. Okay, so the next question for today is, which of the following interactions are required for clonal expansion and differentiation of naive T cells? Is it MHC TCR interaction, B7-CD28 interaction, cytokines binding to cytokine receptors, or all of the above? So you'll have 1 minute for this question and your time starts now. So the right answer is all of the above. So you remember the interaction we were talking about, receptor and co-receptor, to make sure that the cell recognizes self and non-self. Okay, so we'll see a small video with respect to these interactions, and it will help us understand
In
a
this
better
video,
way.
we'll talk about T cell activation. Welcome to Handwritten Biology, where we draw it to know it. So, T cell activation happens in the lymph nodes. But before that, we should have a quick recap of T cell development. T cells are born in the bone marrow from the hematopoietic pluripotent stem cell. Hematopoietic pluripotent stem cell actually give rise to lymphoid progenitor, which eventually produce a immature precursor T cell. After it is born in the bone marrow, it moves to the thymus. It's the training school for T cells. Thymus leads to production of T cells which are mature. They learn all the techniques to recognize peptides from MHC, but still they haven't encountered antigens. That means they are still naive. They have the training, but they really didn't handle real life scenarios. These are naive T cells. From the thymus, naive T cells goes to lymph node, and this is where the action begins. So all the events of T cell activation is actually happening in the lymph node. Now let's talk about the T cell activation. One of the key players that helps in T cell activation is the antigen presenting cell. For example, the dendritic cell. In this case, the dendritic cell has engulfed some pathogens, in this case some bacteria, and now this dendritic cell would display some of its peptides onto class 2 MHC molecule. So the dendritic cell displays some of the peptides in class 2 MHC molecule. Now the T cell, which is naive, which has the T cell receptor, would recognize this class 2 MHC bound peptide. So TCR and MHC complex mediated interaction give rise to the signal 1 for T cell activation. Also, these dendritic cells express CD8086, which binds to CD28 on the T cells. This is the second interaction giving rise to the signal 2 for the T cell activation. Ultimately, T cell also secretes interleukin 2, which in an autocrine fashion binds to the interleukin 2 receptor and give rise to the signal 3 of the activation. So this works like a combination lock. You need 3 digits to be aligned properly for that lock to open. Here all these 3 signals has to be there for the activation of the T cells. If all of these signals are together, then what happens? T cell gets activated. So the second signal was the CD80 and CD86 and CD28 interaction, and the third one is the autocrine signaling by IL-2 receptors. So if any one of these signal is not present, then the activation would not happen optimally. So overall, what we learned that T cell activation requires 3 signals. This is the take-home message. But question is how it is clinically relevant. In subsequent videos, you would understand that, because many aspects of T cell activation are associated with autoimmune disorder. So if someone can tamper around with basic signaling pathway, they might have a solution for these autoimmune disorders. In subsequent videos, we would learn about it. So T cell activation takes place in the lymph node, which is another piece of information which we have to understand. Now, lymph node has efferent and efferent lymphatics. I mean, efferent means exit, afferent means entry point. So all the dendritic cell recognize the pathogen in the periphery, and they migrate to the lymph node, enter the lymph node where T cells are physically located. Now inside the lymph node, there are dedicated areas for T cell and the B cell. Naive T cell and the dendritic cell interaction happens in the lymph node. That lead to activation of the T cells. Now once the T cells are activated, they would rapidly proliferate and divide and increase their number. So they would clonally expand. This is known as clonal expansion. And this is an important phenomena in the process of T cell activation. Eventually, based on what type of pathogen was there, what type of cytokine signaling is available, these particular T cell can become Th1 subtype or Th2 subtype or even Th17 T regulatory, etc. In a separate video, we are going to focus on each of them, and each of them has their own function. All of them has one thing in common: they have the CD4 receptor. So in summary, T cell activation requires 3 signals, which we talked about. You can take a quick screenshot of this and it would be useful for you. I hope this video was useful. If you like this video,
Yeah, okay. So this was the basic detail about how well we have to know the receptor and the co-receptor combination to make sure that our only cell is being activated or recognized. Otherwise it will lead to unnecessary diseases. Okay, so we'll take this last question for today and then we'll carry on with these questions in the next class. So the T cell in the secondary lymphoid organs are assembled by. So this you should know if you have gone through this week's lecture. So I'll just give you maybe 1 minute for this question. It's a pretty easy question. So your time starts now. Yeah, so the correct answer is CCL 119. I might discuss CCL class or CXCL things maybe in a separate class. It completely depends upon how much time is left for our course. And yeah, this was pretty easy. So I request you all to see the lecture videos of the weekly lecture videos, and then only come to the discussions. It will help you understand and refine your concepts in a far better way. Okay, so that's all from my side today. Then we'll meet in the next class. Thank you so much. Bye bye.